Hello. There we go. All right. Hello. Now, this is my critical analysis three discussion. So in these last two lectures, we talked about bacteria, archaea, and fungi. So we know that bacteria and archaea are both single cell prokaryotes. Um, but these prokaryotes can be divided into five different categories, which are obligate aerobes, microaerophiles, facultative anaerobes, aerotolerant anaerobes, and obligate anaerobes. And basically, obligate aerobes rely on oxygen produce in order to produce their ATP and to grow and survive. Microaerophiles, they require oxygen as well, but they require them in lower levels than what is present in the atmosphere. Facultative anaerobes and aerotolerant anaerobes are the most adaptable of these five groups of prokaryotes. Um, the facultative anaerobes, they primarily produce their ATP through aerobic respiration, but they can also switch to fermentation in the absence of oxygen. Um, they can live in basically any environment because they have the ability to go through aerobic and anaerobic respiration. Um, Aerotolerant anaerobes, they use fermentation to produce their ATP. That they use fermentation to produce their ATP, but they can actually protect themselves from oxygen molecules, and they can also live in a wide variety of environments. And then you have obligate anaerobes who use fermentation to produce ATP, and they can only live in the absence of oxygen. And these obligate anaerobes are probably the oldest species of prokaryotes because they never adapted to the presence of oxygen. So we hypothesize that these are the oldest species of prokaryotes because they were around before the Great Oxygenation event. Um, it dates back way back to the early the Earth days. You also have saprobes that feed on dead organic matter. You have parasites that feed on host cells. Um, and you also have photoautotrophs and chemoautotrophs. Basically, photoautotrophs are organisms that use sunlight to make their food. They're typically your photosynthetic organisms, and then your chemoautotrophs are organisms that oxidize inorganic matter such as iron and sulfur to make food. Um, all like bacteria, archaea, fungi, they can all be grouped based on whether they're saprobes, parasites, photoautotrophs, chemoautotrophs. Um, so yeah, basically bacteria, they come in three different shapes. You have rod-shaped bacteria that are known as bacilli. You also have sprout-shaped bacteria that are known as spirilla or spirochetes. And you also have spherical bacteria known as cocci. Um, and your bacterial cells have 70S ribosomes. They use aerobic respiration and produce 36 ATP molecules. Your cyanobacterial cells also have the 70S ribosomes, but they use oxygenic photosynthesis and they also produce 36 ATP. Now, on the other hand, archaea cells, they have 80S ribosomes and they use anaerobic respiration and fermentation, and they actually only produce two ATP molecules, so super low energy. Um, and then you also have groups of bacteria. Um, Bacteria, they can actually be grouped into gram-positive and gram-negative. And that grouping is based on the amount of peptidoglycan and their cell walls and their layers. Your gram-positive bacteria have more peptidoglycan layers, and your gram-negative bacteria have less peptidoglycan layers. And then when you stain these bacteria, the gram-positive bacteria will actually stain purple, while the gram-negative bacteria will stain pink. Um, Archaea actually have different lipids, so we don't group them as gram-positive or gram-negative because their cell walls lack peptidoglycan. Um, it's also important to know most archaea are extremophiles. Um, and then there are also two fermentative processes that bacteria can go through, which are the fermentative hydrogen production, um, and that converts organic substrates into dihydrogen or biohydrogen, which is H2 or hydrogen gas. Um, and they also go through lactic acid fermentation, which is a process that converts glucose or different six carbon sugars into cellular energy and lactic acid as a byproduct. Um, there are also three different ways that bacteria transfer their genes. Um, yeah, transformation is one of them, in which a live bacterial cell actually accepts DNA fragments and genes from a dead or decaying bacterial cell, and recombines that DNA with its own plasmid. And conjugation is another form of gene transfer. Cell to cell contact actually must occur for DNA to be transferred from one living bacterial cell to another. Um, and then in transduction, DNA transfer from one bacterial cell to another is actually done through a bacteriophage. These are typically viruses. The virus transfers, grabs the DNA from one bacterial cell, and then the virus transfers it to the other bacterial cell, and that's how DNA gets recombined. Uh, let's see. So with our next lecture, we actually talked about fungi. And fungi are extremely interesting. I think I would like to learn a lot more about different fungi, specifically different mushrooms. I'm very interested in mushrooms. But the fungi kingdom actually has about 100,000 species of fungi. They have various uses in medicine and food. They're the major decomposers in pretty much all environments that they exist in. Um, and then they also, some strains of fungi are deadly. They can cause different ailments such as athlete's foot. It's pretty disgusting. They destroy library books. They destroy crops. They do a lot of different things, fungi. Um, their fungi are eukaryotic, which means they have a membrane-bound nucleus. They also have organelles, and yeah, they're mostly multicellular. Um, the only unicellular fungi or fungus that we know of is yeast. That's used in beers and wines and bread. Yeah, yeast. Um, and fungi are also like heterotrophic, so they're decomposers and parasitic. Um, so fungi have parts that are extremely unique to fungi. Um, Hyphae are the vegetative bodies of most fungi, and they're constructed of tiny filaments. Um, there are a bunch of different types of hyphae. Um, the first one we talked about was septate hyphae, which are multicellular and contain walls divided by septa. Um, Now, in contrast to that, you have conocytic hyphae. And these hyphae are continuous, like they have a continuous cytoplasm mass, and they're actually multinucleate. Um, they have no septa. So septate hyphae, they're uninucleate but multicellular. Conocytic hyphae are multicellular, are not multicellular, but are multinucleate. So it's one cell, multiple nucleuses, multiple nuclei. Um, and then you have Haustoria, which are another type of hyphae. They're modified and they're found in parasitic fungi. They absorb nutrients from the host, and some fungi have hyphae designed for preying on other animals. Um, and then the mycelium is an interwoven mat of hyphae. Um, and then you have different divisions of fungi. I think there are about one, two, three, four, five. Well, there are five divisions of fungi that we talked about in our lecture. First one was Chytridiomycota. These fungi are actually mainly aquatic, uh, only or mostly found in water. Um, some of the chytrids are saprobes, which are decomposers, um, major decomposers, and then others are actually parasitic, and they're parasites on protists, plants, animals, pretty much whatever, any other organisms in that environment. Um, their cell wall is formed with chitin and flagellated zoospores, and then they're considered to be the most primitive fungi as well. So a lot of different fungi have different ways of reproducing. Um, so with the chytrids, um, they kind of they reproduce with spores, of course, and they have a mature sporophyte that produces spores, and then that will then form a young gamete, or young gametophyte. The gametophyte is what produces the male and the female gametes. Once those gametes, the male and female gametes fuse, they will form another sporophyte, and then the cycle just repeats itself. So it goes from sporophyte to to spores to gametophyte to gametes back to sporophyte. And then your sporophytes are diploid, 2N, while your gametophytes and gametes are haploid, which is 1N. Um, Another division under the fungi kingdom is the Zygomycota division, which predominantly live in soil, water, and food. Some of these fungi are parasitic, can also be resistant to freezing and drying. Um, and then their way of reproduction is different. They actually go through plasmogamy and karyogamy. So basically, with plasmogamy and karyogamy, you don't have male and female gametes. Um, you just have mating types. You have a positive mating type and you have a negative mating type, and those will fuse together. Um, and then plasmogamy happens when the cytoplasms join together, and then there's a mix of genetic information. Um, and then it forms an N plus N individual. Um, Following the plasmogamy is when you get into the karyogamy, and then the karyogamy is when genetic information crosses over and then forms a 2N individual. This will then produce spores. So basically, plasmogamy, N plus N, karyogamy, 2N. Then you have Ascomycota, which are another division of fungi, um, also known as sac fungi, include truffles, yeast, and penicillin. Um, like I mentioned earlier, yeast is the only unicellular fungus that we know of, and also the only fungus that goes through alcoholic fermentation. As I mentioned before, this is used commonly to produce beer and wine and other alcohols. Um, and then you also have lichens, which are basically a symbiotic relationship between algae or cyanobacteria and a fungus. So it can be a fungus and algae, or it can be a fungus and a cyanobacteria. It can be all three. Um, and then they also go through plasmogamy and karyogamy, the Ascomycota. And then another division of the fungi kingdom is the Basidiomycota. Um, these are actually the most common types of fungi, that are club shaped and have the club shaped reproductive structures. Um, these are actually they actually form most of the mushrooms that are edible for human consumption. Um, Most mushrooms won't kill you, but a lot of them are poisonous. A lot of them can have you sick. Um, but a lot of them are edible. We use a lot of them for food. We use a lot of them to cook. A lot of them can be used in herbal medicines, teas. Um, and then a lot of people use mushrooms um, for their psychedelic effects, as a psychedelic substance. So yeah, they also Basidiomycota. They also go through plasmogamy and karyogamy. Um, and then the last division of fungi that we had talked about was the Glomeromycota division. Um, and then the main purpose of these fungi is to form mycorrhiza. And then these fungi actually don't go through the process of plasmogamy or karyogamy. Um, and unique to this fungus is that their reproduction actually occurs in the root. Um, and that's pretty much what we had discussed in these lectures. So thank you.